Hello everyone. This lecture is on Chapter 4, Sections 4.3 through 4.5 in Essential Biochemistry. And what we did in class was to accompany this lecture, we used the Amino Acid Starter Kit from the Milwaukee School of Engineering. So it's a nice accompaniment to this lecture. So we start out by describing 4 levels of protein architecture. There's primary sequence, which is the sequence of amino acids in the protein, and position of disulfide bonds, if there are any. And that's shown right up here for lysozyme. So again, we can see the order of the amino acids from 1 to 124. Oh, sorry, this is a ribonuclease. And the positions of the disulfide bonds. Secondary structure, of course, referred to the alpha helix and the beta sheet, and we talked about both of those last time. So today we want to focus on tertiary and quaternary structure. So what's tertiary structure of globular proteins? Well, first of all, globular proteins, remember, are water-soluble, roughly spherical, highly folded proteins. They're not like the fibrous proteins we talked about last time. There's no pattern to the amino acid sequence. So it's not like the fibrous proteins like collagen that we talked about last time. So myoglobin, hemoglobin, enzymes, those are all examples of globular proteins. The tertiary structure describes the folding of the protein's secondary structural elements and specifies the positions of the atoms. So it's easy to remember because the tertiary structure is the three-dimensional structure of the completely folded and biologically active protein. So this diagram right here is meant to show the folded proteins. So these corkscrews are the alpha helices, and then these arrows are meant to represent the beta sheets. So how do we determine the tertiary structure of a protein? Well, this is how you would do it for myoglobin, say. So if you had myoglobin in solution in dilute buffer, you could add ammonium sulfate. And in 3 molar ammonium sulfate, the myoglobin will precipitate and form some really nice crystals, as you can see down here in the lower right-hand corner. So once you have your crystals then, what you would do is you would put your protein crystal and aim a beam of X-rays at your protein crystal, and the positions of the atoms, they're about the same width apart as the wavelength of an X-ray. So what happens is the protein crystal acts as a diffraction grating. So if you have a piece of film right here, you will see dots where the X-rays have exposed the film, and then you will be able to, if you're a crystallographer, which I am not, you'll be able to work backwards. So you would produce something like this, and if you have if you know what the pattern of the dots are, you would be able to sort of work backwards and figure out the structure of the protein that produced that diffracted image. So here's an electron density map here. So there are, of course, powerful computers that will help us go from electron density maps and be able to map the primary sequence onto the tertiary structure. So different images might have different resolutions. So the lower the angstrom resolution, the sharper the image. So if this is the structure of a protein here, you can see over here on the right we have a nice sharp image, or over here the image might be a little blurry, sort of like a blurry photograph. This occurs because getting protein crystals is a difficult task. Protein crystals aren't like sodium chloride crystal. The proteins are actually highly hydrated and jelly-like, so sometimes that limits the focus or the atomic resolution of the crystal structure. So if you have some knowledge of the primary amino acid structure, that can help you with the resolution of your tertiary structure. So the way we visualize protein structure, we've already been doing this somewhat, but we want to point things out here. You could have something called a ribbon diagram. So here, we're just following the polypeptide backbone. The side chains of the amino acids aren't shown here. So we can see the alpha helices, and then here we can see a core of beta sheets, again shown here as arrows. This is a triphosphate isomerase. So that's one way. This structure might give the impression that there's a lot of empty space in proteins. There isn't. If we have the space-filling models where we account for the electron clouds of each of the atoms, we have something that looks like this. So this conveys to us that these proteins are highly folded, highly packed, but it makes it difficult for us to see those secondary structural elements. We might just want to have a trace right here where, again, we just look at the polypeptide backbone. So there are lots of different ways of visualizing protein structure. So, so this is for ribonuclease. This is the primary sequence right here. So again, we defined earlier the primary sequence is just the order of the amino acids. Again, so here I know the order from 1 to 124. I know the positions of the disulfide bonds. But this image just tells us the primary sequence, and that's all it tells us. This is the tertiary structure. So here we don't see the sequence of the amino acids, but we can see the positions of the disulfide bridge. We can see that this protein has some regions of beta sheets, and it also has some regions of alpha helices, along with some coiled structures or just random structures here. Okay, so that's tertiary structure. What about quaternary structure? Well, not all proteins have them, but some proteins consist of 2 or more polypeptide chains. So this diagram over here represents that, where we have this shows 2 chains that are associated to form the whole protein. So it's important to note that if we take these subunits, these chains, and pull them apart, the protein wouldn't function. So in order for this protein to properly function, all of its subunits have to be present, and they have to be organized in a way that allows them to function. So the language we can use here is we can say that this protein has 2 chains. We can say it has 2 monomers, 2 subunits. Any of that language would convey that we have a protein with quaternary structure. So here's some examples from our book. So here the artist has drawn each one of these chains in a different color. So one of the things that we should keep in mind is that what keeps these subunits together is typically they're associated non-covalently. So if we look at this protein right here, the hydrophobic effect is still very much in play here, and the interfaces of these individual monomers will have hydrophobic amino acids because they are literally in the interior of the protein. Hydrogen bonds and ion pairs or ionic interactions would also be important in keeping these together. In some cases, there are covalent bonds. There might be disulfide bonds that keep subunits together, but typically subunits associate non-covalently. So to summarize, what's primary structure? It's the amino acid sequence in a polypeptide chain, like shown here. What's secondary structure? Well, those are these secondary repeating structural motifs, like the alpha helix, which is shown here. What about tertiary structure? Well, that tells us the folding of the three-dimensional structure of the completely folded protein. And quaternary structure, so of course this is shown for hemoglobin. Hemoglobin has 4 subunits that come together to form the functional protein. So a nice way to show the concepts of protein folding and stability is to use the Amino Acid Starter Kit from the Milwaukee School of Engineering, and that consists of a long tuber with clips that you can put the amino acid side chains. So what we did in class is this tuber had a blue end, a blue cap, that was the amino terminus, and students chose 6 hydrophobic, 2 acidic, 2 basic, 2 cysteine, 1 methionine, and 2 other polar side chains to make a long chain that was 15 amino acids long. So while we tried to do this exercise, we also had a conversation about the forces that stabilize protein structure. The hydrophobic effect is the major effect in stabilizing protein structure. Electrostatic interactions are important, as are van der Waals interactions. I prefer to call them London dispersion forces because different textbooks define van der Waals interactions different ways. We might also have chemical crosslinks, metal ions, or hydrogen bonding. All that is a minor stabilizing force, since you could easily form hydrogen bonds with water in an unfolded protein. So let's talk about the hydrophobic effect first, because that is the driving principle in protein folding. So the model we made in class looks a lot like this, where you have this tube right here, which represents the polypeptide backbone, and all of these little circles here are the side chains. So here the artist has represent nonpolar side chains in green, whereas there are a variety of different polar side chains shown in yellow, red, and blue. And when the protein folds, all of the green nonpolar side chains are relegated to the interior of the protein. Remember, the hydrophobic effect says that water refuses to interact with the nonpolar side chains. Water doesn't form favorable interactions with the nonpolar side chains. So this protein folds, so we have this hydrophobic core with our nonpolar side chains, and then we have this variety of polar side chains right here. Each of those red dashes is meant to represent a favorable interaction with water. So again, just to remind us, the hydrophobic effect describes water's refusal to interact with the nonpolar amino acid side chains. So they will interact via weak London dispersion forces in the middle. So again, they are there because water refuses to interact with them. So these nice drawings by Irving Geis can illustrate this too. This is horse heart cytochrome C. It has a prosthetic group that has an iron right here shown kind of as a sun, and this is an organic group right here. So the iron plus this porphyrin ring is called heme, and the polypeptide backbone is shown in blue, and the amino acid side chains that are hydrophilic and form favorable interactions with water are shown in green. And you notice that they are largely on the outside. Occasionally we'll have some exceptions. For example, you can see that's a histidine right there that forms an important interaction with the heme. And sometimes you'll see that in proteins where you'll have in the interior, in the cavity, there will be polar amino acid side chains that have important structural and functional roles. Particularly with enzymes, you will see this. But mostly you find the polar amino acids on the outside. If we draw this again, here the red and the orange amino acid side chains are shown. These are the nonpolar side chains, and again you can see that these are largely on the interior of the protein. So these drawings are a nice illustration of protein folding via the hydrophobic effect. So when we had our tubers in class, I showed you the diagram where the tuber is folded, so its shape is consistent with the hydrophobic effect. And again, this is from our textbook, if you have, say, this green region right here with that consists of nonpolar amino acid side chains, the protein is going to fold so that the nonpolar amino acids are on the inside away from water, because water does not interact with it favorably. And then the orange over here represents polar amino acid side chains that can form favorable interactions with water. So you might think that this violates the second law of thermodynamics, because this folded structure certainly is more ordered than the unfolded. But remember, you have to think of the entire system, and you have to think about the water molecules. So water molecules would form ordered cages around the nonpolar side chains if you forced them to interact with water. So the entropy of the entire system is increased here because the water molecules are far more disordered in the presence of a folded protein than they are with an unfolded protein. So this is illustrated here. I found this article, and I have my reference down here. It's a little unfortunate that whoever made this looks like they used a Word document and had the function on where you can see the spell checker and the paragraph marks. But anyway, I do like it because it shows an unfolded protein right here, and it shows the backbone in green. It shows the hydrophobic groups that are in yellow. And it shows very, very ordered water molecules. Now, if you have a carbonyl group, these interactions will be favorable. And these, if you force them to take place, would be very ordered. So when the protein folds, so that the nonpolar amino acid side chains are in the interior, notice that you have a very, very disordered population of water molecules surrounding the protein. So this over here, a folded protein really is thermodynamically favorable because this system over here really is more ordered. Here, the folded protein with the highly disordered water molecules, the system there is more disordered. So let's look at forces that stabilize the protein structure. So we have the hydrophobic effect, so we were able to fold our protein. Now we have some other amino acid side chains that we're interested in looking at here to look at the concept of electrostatic interactions. So in the old literature, these could be called salt bridges. So the current form is ion pairs. So if you have a lysine, which is typically protonated at physiological pH, and a glutamate, which is negatively charged, you have a nice ion pair here, or maybe a positively charged arginine and a negatively charged aspartate. Now, one thing I need to challenge you to remember, just like when we looked at our water molecule kit a few weeks ago, these positive and negatively charged side chains are highly hydrated. So if you want to form these interactions, you will have to strip off the water molecules in order to have this interaction occur. So if that is going to take place, that means that the ion pair interaction, it must be worth it, that there must be some stability offered by that was offset by the amount of energy required to desolvate those chains. Okay, so the London dispersion forces, so remember those nonpolar amino acid side chains ended up in the interior as a consequence of the hydrophobic effect because water refuses to interact with them. These London dispersion forces are weak. They act over short distances. They are still significant though, and you do lose those interactions when the protein is folded, the protein is unfolded. Other forces that could stabilize protein structure, this is sort of a generalization, but in extracellular proteins you are more likely to find proteins that have disulfide bonds because we have an oxidizing environment. So forming the disulfide bonds requires that we lose hydrogens on the cysteine side chains to form the side chain, and you typically don't find these in intracellular proteins because reducing conditions exist there. Some proteins are stabilized by metal ions. Not all, but here's an example of something called a zinc finger. They're called that because if you look at a two-dimensional drawing, they kind of look like fingers. So these purple balls right here represent zinc ions, and they're not shown in this diagram, but you'd form a coordinate covalent bond between amino acid side chains that either have nitrogen or sulfur. So these are cysteines right here in yellow, and these are histidines right here with the blue nitrogen. So here's an example of how one zinc finger would look like, and here's another one where you've got two zinc ions and a number of cysteine side chains that form the coordinate covalent bonds. So let's not forget hydrogen bonds. Again, they are minor, and that's because the hydrogen bonds between amino acid side chains, you could easily form them with water if you unfolded the protein, but they are still important in a folded protein. So if we look at the green, this is a polypeptide backbone right here, and there are lots of different places where you can form hydrogen bonds. Remember, the peptide group has the capability of forming hydrogen bonds. So between two peptide bonds, between a peptide bond and amino acid side chain, or between two amino acid side chains. So this table illustrates that in a little bit more detail. So for example, if I had two amino acid side chains with hydroxyl groups, here's my hydrogen bond donor and my hydrogen bond acceptor. Maybe a hydroxyl group right here, and this could be a carbonyl and a peptide bond. So again, hydrogen bond donor, hydrogen bond acceptor. Maybe an amide and a carbonyl. Again, you find both of these in the peptide group. An amide, that you find that in the peptide group, maybe a hydroxyl over here. Here's an amide, and there's a histidine. So there are lots of other examples, of course. So this is the typical distance between the donor and the acceptor in nanometers. So if they're farther apart than that, then you wouldn't be able to form a hydrogen bond. So here's some picture. This is obviously in the before times because no one's wearing a mask. But what you see here is that I gave the students the directions to fold up their proteins, and all of them looked different, even though supposedly everybody followed the same directions. So what I told them to do is I told them to fold the proteins so the nonpolar amino acids were on the inside and polar chains were on the outside. Some of the students were able to take negatively and positively charged side chains and put them together to make an ion pair. They could take two cysteine residues and form a disulfide bond. But the three-dimensional structures weren't the same, and the reason for that is that I never specified the order of the amino acids. So when the students put the amino acids on their tuber, each of the groups would have had a different amino acid sequence. So that points to the importance of the amino acid sequence in specifying the structure of the protein. So now that we put our protein together, the next thing we wanted to do is we want to take it apart. So the word that we use here is called denaturation. So when we denature a protein, we unfold it. And if we unfold the protein either completely or even partially, the biological activity is going to be lost. And in vitro and in vivo, when a protein transitions from folded to unfolded, it's a pretty dramatic transition, meaning that the protein unfolds cooperatively. That means that once the unfolding process gets started, then it happens very rapidly after that. So how do we denature a protein? Well, there are lots of different ways. We could add a reducing agent, so beta-mercaptoethanol or dithiothrietol. These are agents that can donate hydrogens to disulfide bonds to reduce them to sulfhydro groups. So if a disulfide bond is important in stabilizing your protein and you reduce it to sulfhydro groups, then you'll no longer have that stabilizing force. How else can we denature a protein? Well, we could heat it up. This would increase the vibrational and rotational energy of the atoms, which disrupts weak interactions. We could break the disulfide bonds, if we haven't broken them already, with our dithiothrietol or beta-mercaptoethanol. We could deamidate azin and glin to asp and glue, respectively. And if those amide side chains were important in hydrogen bonds, then we would lose that stabilizing force. And speaking of that, if we raise or lower the pH pretty dramatically, we can change the deprotonation of the side chains. Maybe you protonate something that shouldn't be protonated if you lower the pH, or you deprotonate if you raise the pH. And again, if any of those functional groups are involved in hydrogen bonds, you would break those bonds. Or maybe if you protonate or deprotonate a functional group, you could disrupt an ion pair. How else? Well, we could add a detergent. So detergents, as we talked about in Chapter 2, they have a nonpolar domain, and that's able to wiggle into the interior of the protein, and it interferes with the hydrophobic interactions responsible for the protein's native structure. So that's how detergents are able to unfold proteins. And finally, we can add something that's called a chaotropic agent. So think chaotropic, think chaos or disorder. So guanidinium hydrochloride and urea, the structures are shown here at the bottom. It's not exactly understood how they unfold proteins, but probably it's similar to what detergents do, where you disrupt the hydrophobic interactions. So I want to tell you about Christian Unfinson's experiment. He added urea and beta-mercaptoethanol to ribonuclease, and as I showed you at the beginning, ribonuclease has this folded structure, a single chain, 4 disulfide bonds, and if you add 8 molar urea and beta-mercaptoethanol, all of those disulfide bonds get reduced to sulfhydro groups, and the protein is unfolded with urea, the chaotropic agent. So you lose your tertiary structure, and the biological activity is lost too. So ribonuclease is an enzyme that hydrolyzes the nucleotides in an RNA. So the ribonuclease isn't going to work anymore if it's unfolded. So Anfinsen showed that he could reverse this process. So he removed the urea by dialysis, and it's actually fairly easy to oxidize a sulfhydryl group. You just expose it to oxygen, and you can oxidize these sulfhydryl groups and reform the disulfide bonds. So fairly miraculously, the protein was able to fold right back up into its native conformation, and the biological activity was restored. So this illustrates the importance of primary sequence in dictating tertiary structure, and our model exercise showed us that too. That if you have a primary sequence that that's different, you can get a different tertiary structure. And if the protein is able to refold into its tertiary structure, there must be some information in the primary sequence that allows it to do that. So it was proposed that proteins fold spontaneously in a cooperative, sequential, and non-random way into their native conformation, maybe seeking the most thermodynamically stable structure. But that isn't really possible for a protein to sort of try out every possible conformation and then sort of settling on the most stable one. That there really isn't enough time in the cell for that to happen. So much after Anfinsen, his experiment I think was in the late 1950s, and proteins called chaperonins were discovered in the 1980s. So there was a pretty big gap in between here. So in the cell there are proteins called chaperonins that assist in protein folding. So if you start out with an unfolded protein, the role of the chaperonins isn't shown here, but they assist the protein into folding into the secondary structural elements first, and then you can see sort of like the tertiary collapse right here, where we get the highly folded protein at the end. This is really important for newly synthesized polypeptides to fold. If they fold properly with the assistance of the chaperonins, then they can assume their native structure, which is biologically active. If they misfold, the chaperonins can assist in the refolding process. If there isn't any way to fold the structure properly, there's a danger that it could aggregate. So there are mechanisms that allow for its degradation, because if the protein aggregates, that could lead to a disease state. So there are actually diseases that are associated with protein unfolding. So I have a protein folding movie that I'm going to show you. So hopefully this works on this recording. I wanted to get out of the ad, so this is from the Pandy lab. So this shows, at the beginning you saw the alpha helix form. Okay, this is the hydrophobic collapse that I showed you. So we have the hydrophobic amino acid side chains are in the center. And now it's stuck in a non-native trap state, so it can't continue to fold unless it folds properly. So the non-native contacts break. So now we're going to form our clean secondary structural units. So that's the beta sheet over here, but we're not finished yet. The third part of the sheet is missing. So if you look over on the left, the C terminal is going to swing in. So watch the C terminus. And then there's an instant replay. Let's see that again. There's the C terminus, and it swings in to form the beta sheet. And now our protein is folded. It locks in with hydrogen bonds and side chains to yield a folded structure. All right, so let's close that out and go back to our PowerPoint. So protein folding is important because there are protein misfolding diseases that we should be aware of. So maybe you knew of some of these, or maybe you didn't. So Alzheimer's disease involves an amyloid beta protein that's defective. ALS is due to superoxide dismutase. Huntington's disease, there's a protein that has a mutation where there are lots of polyglues in a row, and that means the protein can't fold properly. Some of these other diseases maybe you haven't heard of. Parkinson's, maybe that's due to a failure of the alpha-synuclein to fold properly. And the transmissible spongiform encephalopathies, those are the prion protein caused diseases. So these are things like mad cow disease, or in cows, and scrappy in sheep, and the Jakob-Creutzfeldt disease in humans. So that is my protein folding lecture. So thank you for listening. Thank you for your attention, and see you next time.